Well, Simon, I'm in London. I really enjoy to meet you. Well, you are in the most amazing field for me for Gen AI. I want just to explain your transition to this field because at the first you studied physics. Is that right?
Yeah, that's right, Pablo. So my name is Simon Cole. I'm the founder and CEO of Latent Labs, where we work on generative models for biology. We're starting in the area of protein design. So really, we're working on building these frontier models that generatively make new proteins that aren't found in nature with functions that relate to therapeutic effect, for instance. And really, our mission is to make biology programmable and make this technology accessible to others, including companies and academics out there. Really, I think we're at a fascinating, juncture in the road of science where we're seeing AI have a real impact on the life sciences and science beyond. So, personally, actually, my science roots go way back. Like Pablo just said, I studied physics, then did a PhD in machine learning and AI, applying and building models in the area of medical image analysis, and then moved on to DeepMind, where I first interned in the health team, and then actually joined the AlphaFold team there. So I was part of the AlphaFold 2 team from the start, and including worked on different aspects of the model, core pieces of the architecture, including the uncertainty predictions. And as perhaps you're well aware, AlphaFold 2 made quite the splash. So it won CASP 14, rivaled Experiment in protein structure prediction with an AI model. And then, of course, recently in December of last year, was awarded, if you will, the Nobel Prize in Chemistry that was given to Demis Hassabis and John Jumper. And so really we're at this.
That's amazing. That's amazing. Just you are in the team with this Nobel Prize in Chemistry, and just to explain a little this first stage of the artificial intelligence with AlphaFold that you need to figure out how about the proteins.
Yeah, that's right. So AlphaFold 2 really is a model that from the amino acid sequence predicts its structure. And amino acids are these building blocks of life. Our human bodies and really any life forms are largely made of proteins. So they're the building blocks. And you can think of them as sequences of amino acids. And amino acids come in 20 different varieties in the human body at the least. And so it's sort of these strings of 20 different letters that make up proteins. And these sequences, they fold up in space in characteristic ways and form a structure. And it's this structure that gives them their function that sort of determines how they interact with other proteins and what they do in our bodies. And so, for a long time, biologists have been very interested in the structures in order to understand function better. But that had required a lot of time and effort, many times futile actually, to determine these structures via experimental ways. And so it's been a long-standing problem in biology to essentially circumvent that, trying to predict from the sequence alone the structure of these proteins. And that's what AlphaFold 2 has achieved, rivaling Experiment in this multinational, international, I should say, competition that's called CASP. Where we entered and really showed that we're able to match experimental performance for single chain protein structure prediction.
Was like a long journey to do that, no? Because all the scientists try to predict these structures, and the mathematician problems around that is totally complex.
Yeah, that's right. It's been a long time in the making. CASP, that competition I mentioned, has been going on for decades, and so it's been a really long-standing problem. And it's very important for me to also stress that we were, as a team at DeepMind, building on sort of a lot of prior work. The way the problem was evaluated, the way that data was collected that we could use, the way that certain ideas had been around in the field that we were able to build on top of. So it's sort of in that sense, been able to sort of really build on top of the shoulders of giants, and then made this breakthrough in terms of what was possible with AI here.
And the nature discovered some proteins. I don't remember the exact number, like 4,000 probably you know where, but you can build a lot of more, like a lot of magnitudes more proteins that was already created by nature. And this was Gen AI come into the field because for us, for the normal people, Gen AI is like language. It's just to discover the language and and you don't have an idea of how artificial generative intelligence make a role here. No, it's like the protein has like an alphabet also, and you have some syntactics to go to the semantics as we do with the language.
Yeah, so proteins in nature are abundant, as I mentioned, but it turns out that nature itself sort of clearly hasn't sampled at all the entire space of possible proteins. Actually, a tiny, minuscule subsection, if you will. And really, you can think of AlphaFold 2 as a model that allows you to more efficiently visualize these protein structures by predicting them. So in that sense, it sort of acts like a microscope, if you want to use that metaphor. And really, it allows you to visualize and predict structures of existing natural proteins. But then, as I just said, there's this huge white space of proteins that nature hasn't sampled, hasn't sort of gone into testing, if you will. Mm-hmm. And with generative models, having learned the fundamentals of biochemistry, we're now pushing the boundaries there, going beyond designing proteins from scratch where necessary. It's not always necessary, but we can do that now. We can bestow proteins with new functions, and really that's what we're working on at Latent Labs. Generative models that are going beyond what nature has discovered.
And that's why your company have a lot of attention, and you really have a lot of funds. I try to talk about that also, because I also you to mention these two persons that are very important persons in the history of AI. One of them are the author of the Transformers paper, The Attention is All You Need. That is one of the main papers of the history of Gen AI. And then they give their money to you, not just as a VC, also as a person.
Yeah, that's right. No, we're very lucky that recently we were able to raise a Series A. In total, our funding has gone up to $50 million with that, which really equips us to build out all the things that we're setting out to do with lots of momentum. And as you said, a fantastic group of people and investors have come together to support us. So on the one hand, it is institutional investors, like you mentioned. We have a great team of co-leads in Radical Ventures and Sofinnova Partners that sort of have given the bulk of the funds. But then equally, like you said, amazing scientific individuals have contributed. For instance, the Transformer paper co-author Aidan Gomez, or the 11 Labs CEO Mati Staniszewski, and even Google's chief scientist Jeff Dean. So we have really a fantastic group of people around us.
Wow, it's amazing. And the work you are doing to discover these new proteins, these like large language models for biochemistry, or for is the same the procedure with Gen AI. We have like 44 terabytes of data of the internet, and well, this with embedding these words to figure out some connection between the words. Well, it's a lot of mathematics around that, and then some syntactics emerge, and then some semantics also. One was amazing. So you have this also huge database of proteins, and you are working like SML with little ones first, or the approach is like a huge one at the first.
Yeah, machine learning at a really high level, you can look at it as learning the hidden patterns that are in the data and that are otherwise really difficult to capture in a rule-based, maybe in a simple math-based fashion. That's where they really shine. Of course, as you would know, they're these sort of fantastic composed architectures that are trainable and sort of really highly flexible to extract that underlying system of patterns from the data. And in that sense, you can really look at deep learning as a compression of the data into model weights. And that's sort of the process of learning, really. And, as you mentioned, right, there have been plenty of breakthroughs over the last couple of years, particularly in generative AI, where, we're seeing models work really well for text, for chat, for images, for video, for audio. And all of that sort of powered by similar paradigms. And the same is true in biology, really. We're doing related work. We're also building generative models with sort of bespoke insights, with bespoke elements in terms of the architecture and how we sort of treat the data. But at the end of the day, again, it's compression, right? We have data available for the sequence and structure of proteins, even beyond different molecular types are of high interest to us, like DNA, RNA, small molecules, and how they all interact. And there's a lot of great public data sets out there, which again, huge credit to the folks who have painstakingly often collected them and made them available. So that's why this is possible. And yes, we're training on large scales of data. We're able to build multimodal models, which help us leverage different sources of data. In many ways, you could describe that as multimodal because that gives you a lot of flexibility in terms of what type of data to learn from. We're largely sourcing data out of the public, but then a lot of work is put into curating, cleaning, augmenting, enriching that data, and sort of we call that lifting that data out of the public domain such that it's most useful for our models.
And the two technologies could be coexist. For example, with GenAI, we discover some protein, and with AlphaFold, we discover better the functionality around this new protein, or are two separate worlds?
So I think your question probably aims at how our models sit against AlphaFold, and it's this idea of AlphaFold being a predictive model that can, given a natural sequence, predict the structure that corresponds to it. But AlphaFold's not a model that goes beyond in that it's not generating new proteins in let's say a conditional way or similar. And so we're building a whole new class of models that are finely steerable and conditional on requirements that matter in the real world. For instance, properties that are important for drugs, we can bake them in from the start by appropriately conditioning our models.
And what do you think about the first stages, with the first impact that we can see in the near future and in the large future, like science fiction? I mean science fiction, I mean like a prompt, building this protein to make some skin blue or whatever, no? This like science fiction. But we can do this pattern to have first something that that is possible in the next 2 to 3 years, and well, later we see.
Yeah, well, we hope to accelerate and augment drug discovery in the very medium term. We hope that what we're able to provide for in terms of the technology will allow people to do new things that have been traditionally hard. And I think that's going to unlock a lot of impact. It's going to accelerate drug discovery. We're also hoping it will allow us to come up with new modalities, drug diseases that were previously considered undruggable. So very tangible impact that I think in the medium term we think we will be sort of providing. And then otherwise, the sort of dream really is to make it fully push button, to allow to steer every aspect computationally, and really sort of sidestepping what is otherwise an experimental or iterative process.
And you think will be like a ChatGPT moment in the future, like suddenly these models do a lot of things at the same time? Could be. I know it's sure not.
Yeah, I think we're steering towards that. I think there'll be a lot of very exciting progress in this area.
Well, Simon, thank you so much to invite us to visit you here in your office. You have office here in San Francisco, we must say. And well, I hope your team have a very success in the near future.
Thank you very much, Pablo. It was a pleasure to speak to you.